Hello, my dear students. Welcome to our today's theory session. So, in today's theory session, we have to see that what is meant by gene, what are the different functions of gene and role in animal genetics, gene actions, gene and genotypic frequencies. Here myself, Dr. B. N. Thorat, Assistant Professor, Department of Animal Husbandry and Dairy Science. Before going on gene or terminology on gene, we have give some brief information about the gene. That means all living beings have genes. They exist throughout the body. Genes are a set of different instructions that determine what the organism is like, its appearance, how it survive and how it behave in its environment. Then a gene is a basic physical and functional unit of a heredity. Every person has two copies of each gene, one inherited from each parent. Most of genes are the same in all people, but a small number of genes, that means less than 1% of the total population, are slightly different between the people. Genes are made up of DNA, that means deoxyribose nucleic acid. Each chromosome contains many or number of genes. So, who discovered the terminology gene? We have already explained in our history and concept of animal genetics. So, who is the scientist? They have worked or coined the term genes. So, Johnson in the year of 1909, they have coined the term gene. So, we have moved our most important terminology, that is gene. So, gene means it is the unit of heredity which are transmitted from one generation to next generation. Okay, you are clear? Oh, then another terminology or another definition of gene, a portion of DNA which occupying a specific place on a chromosome through which inheritable characteristics are transmitted to the next generation in all living organism is called as gene. So you can write down another terminology, that means gene is the functional and basic unit of inheritance, and these are arranged in a linear order on the chromosome. So again another terminology of a gene. So gene may be defined as a hereditary unit which reside in a long DNA molecule, the main constituent or component molecule of chromosome, and contains coded instructions or information for the production of protein which ultimately causes and determine a phenotypic traits. All of you are remember these different types of different terminology you have to be remember. So these genes are self-duplicating and sometimes undergoes mutation. Gene also control the physiological and biochemical action which are going in the cell. The DNA. DNA is a long coil double stranded molecule which encoded the genetic information. There are four types of bases in the DNA, that means deoxyribose nucleic acid, adenine, thymine, guanine and cytosine. The order of bases in DNA strand, which is called as a sequence, that creates a code for information. The DNA code ATC has different meaning than the code TCA and so on. Each cell is equipped with a special machinery which is used to read the sequence and use the information. What are they made of? The gene consists of a long combination of four different nucleotide bases or chemicals. There are many possible combinations. The four nucleotides are adenine, cytosine, guanine and thymine. Adenine denotes A, then cytosine C, guanine G and thymine T. Different combination of these letter AC, GT give people different characteristics. For example, a person with a combination of AT, C, G, TT may have blue eyes, while the somebody with the combination of AT, CG, CT may have brown eyes. Are you remember? Then this diagrammatic representation shows there are or the bases pair in the DNA. See, this is the chromosome. Okay, so chromosome have also different names of different organs. The middle one, the centromere. Then these are the two arms, P arms and Q arms, and lastly center or telomere. Then this is the double helix standard or double helical standard of a DNA, and this is the chain of DNA model. So this slide shows, this is purple color shows chromosome, and this link or chain of a chromosome, it should be create or replicate a gene. Then again this diagram shows, this is nucleus. In this cell there are different chromosome, nucleus, and these chromosomes are made up of this nucleotide or double helix stand, that is deoxyribose nucleic acid. Then this chain is a DNA double helical standard. Then this chain or link, that is called DNA molecule. Okay, then again there are different colors, they have to be shown in the basis of DNA. So red color shows the guanine, then yellow color cytosine, green color thymine, and white color is a adenine. So this is the diagrammatic representation of their different bases or their 4 bases in the DNA. Again, gene provide information for the synthesis of various enzymes and protein on the body. The protein that are determined by the genetic DNA result in the specific physical traits such as the coloration of an animal coat, the texture of person's hair. We have black color. Okay, that is the color, and another texture, rough structure, smooth texture, etc. The different form of a gene is called as a allele, or allele is the alternative form of an gene. There are at least 2 alleles of a gene, one of which may be dominant over the expression of recessive gene. Okay, so which are the different chemical bases of heredity? DNA, there are 4 bases. In that, first, DNA carries genetic information, then duplication of DNA, then third one, molecule basis of mutation, and last one, mechanism for the translating the genetic information into action. So we have to be move first, that means DNA carries genetic information. In that, the property of information that can be stored clearly lies in the order of purine and pyrimidine bases in the DNA molecule. 4 types of bases are there in DNA molecule, and each gene is said to contain 1000 to 1500 different pairs. Thus, there are at least 41,000 different ways in which these 4 bases can be ordered into sequence. The capacity to store information on a gene is enormous. Then second mechanism, that is duplication of DNA. The property of exact duplication is the result of complementary strands of the double helix. In structure of DNA molecule, each base will be paired with only one other base, that is adenine with the thymine and guanine with the cytosine. Then third one, molecule basis of mutation. All of you are known or heard the terminology of mutation. That means mutation is the sudden change in genetic makeup of an organism that may be passed on homologous chromosome is called mutation. Then last one, that means mechanism for the translating genetic information into action. This genetic information encoded in DNA that was found to contain a sequence of amino acid in specific protein. Then it was discovered that RNA molecule that should be copied the base sequence of a DNA, which can move to the ribosome and provided the template on which protein molecule are formed. Therefore, the phenotypic response is governed by the sequence of bases in the DNA which are coded for the formation of specific proteins which can be responsible for the catalytic reaction, the chemicals in the cell. So these are the mechanism of heredity. Then another most important part in this, The question always asking in your semester examination, what is meant by gene and what are the different functions of gene? So we have moved our focus on functions of gene. So which are the functions of gene? So gene carry genetic information from one cell to another cell, one generation to another generation. Then another gene must be duplicate. That means it can be work as a template for copies of itself. Then gene must be mutate. There must be exist in some mechanism for translating the genetic information into the action. In the developing individual, genes are coded by specific DNA sequence. Then another function of a gene, that is RNA production. Through the process, that means RNA production, is called transcription. DNA is transcribed into the RNA, that means direct product, okay, which is a template for protein production. Then another most important function, that is protein production. This is another process, okay, this is called as a translation, and RNA is translated into the protein. That means RNA is translated into the protein, is called translation. Then last, expression of a character. In theory, one gene coded for one protein. Okay, protein carry out most of the necessary functions in the cell. So these are the different most important functions of DNA that means play in the gene. So you can remember that is the terminology transcription and translation. Translation means RNA translated into protein. Okay. Then which are the different role of a gene in a animal genetics? So in animal genetics, this gene have play important role in the production of enzymes, normal hemoglobin level, then antibodies production, then hormone production, as well as improvement in the performance of livestock, as well as detection of various viruses and production of a vaccine. So this is the one part of gene and gene functions, their mechanism and their role. Okay, another part, that means the gene action and types of gene action. Always asking in your examination what is meant by gene action, but most of the students are confusing in that terminology. So what is meant by gene action? Gene action means it is a way in which certain genes exert their effects on the body. They could be dominant or recessive, or they could be sex linked, or to be involved in the chromosomal abnormality. Okay, the combination of such gene action result in the observable phenotype of an organism, that is known as a gene action. All of you are remember this terminology, gene action. So how their types, or how they are divided into different types. So there are three types of a gene action. So which are that? Gene action, that first, that is additive gene action. In additive gene action, what is working, or what is meant by additive gene action, how they are impacted or how they are play role in the gene action. This is the type of gene action whereby each of the two alleles contribute equally to the production of qualitative phenotypes. Neither allele is dominant. Okay, the heterozygous genotype produces the phenotype that is intermediate between those produced by the homozygous genotype. This action of gene is said to be additive when one allele of a gene is substituted by the another allele and it is produce the same effect. That may be positive or a negative effect, and this can be happen only if the allele is not dominant or recessive. Out of that, both neither be dominant nor be recessive. Another, dominant gene action. In dominant gene action, the type of gene action whereby one allele is expressed more strongly than the other. There are two types, that is complete dominant gene action incomplete dominant gene action, and another in that part, that is over dominance. Okay, so when a dominant gene, we can be given A alphabets, is substituted by a recessive gene, that is small a alphabets, and there is a no effect, either positive or negative. It is called dominance gene action. Then over dominance, in that over dominance, phenotypic value of a heterozygous is more superior of the either of two homozygous. Then epistatic gene action. In case of epistatic gene action, a type of gene action where one gene suppresses or alters the qualitative phenotype which can be produced by a second gene or another gene. This interaction usually result in the production of new qualitative phenotypes. In this type of gene action, the expression of presence or absence of an allele in one locus which can affect the expression of another allele in a different locus. These epistates can either exert additive effect or dominance. In case of, or in other words, we can be explained this epistatic action of a gene can be completely mask the effect of another gene, or it can also function in compulsory unmasking the effect of gene that remains dormant by default. Then third part of the topic, that is genotype and gene frequency. So genotype frequency means number of genes which are present in the population, and it always another terminology, that is gene frequency. That genotype frequency and gene frequency, that part we have already covered in our practical session, and there are different problems on the basis of genotype and gene frequency. But here some theoretical part. Another gene frequency. So gene frequency likewise genotype frequency. Only you can remove genotype words and place gene. That means number of genes which can be present in the population. And this gene frequency is always expressed as a percentage or proportion. Then here some problems that have the formula, they have to be given gene frequency. Okay. So gene frequency, that means number of a gene which is present in the population. So it can be denoted by two alphabets, that is capital A and small a. Capital A frequency, that denotes or shown the number of copies of capital A allele divided by total number of alleles of that particular genes in the population. And small a alphabets or small a genotype, this work as a genotype. The number of copies of allele divided by total number of allele of that gene in the population. And in both cases, both the gene and genotypic frequency, it should be range between the 0 and 1. Then there are some factors which are affecting gene frequency. In that, I have listed 4 most important factors. The first one, selection. Another one, migration. Then mutation and genetic drift. So on this, always asking a different type of question on this topic. That means the first one, define gene and write down their functions. Then second one, explain the chemical basis of heredity. Then another type of question, that is enlist different types of a gene action and explain in detail any one of them. And last one, describe in short factors affecting on the gene frequency. Okay, so this topic is also important. Thank you for your patience. Thank you all of you